investigation of exported protein candidates in Cryptosporidium parvum. Jennifer.
I'd like for you to imagine yourself in the University of Georgia football stadium on game day. And when you look around the crowd, instead of seeing the usual faces of the fan base, you see the faces of nearly 100,000 children. That's the number of children who die from the parasitic infection caused by Cryptosporidium each year. Or to put this into more simple terms, that's the equivalent of one school bus full of children who die due to this parasitic infection every day. Yet many of us here have never even heard of it. Cryptosporidium is a major cause of diarrhea worldwide. And while it's typically a disease that's associated with poverty and malnourishment, it actually can be found right at home here in the United States. As back in 2014, we actually experienced our own small outbreak right here in Athens, Georgia. This is in part due to the fact that the infective stage of Cryptosporidium is resistant to chlorine and other water treatment strategies. A person becomes infected with Cryptosporidium when he or she accidentally eats or drinks food or water that's contaminated with the infective stage of the parasite. So once inside of the human body, the parasite travels down to the small intestine, where it begins to make a home for itself. It's thought that the parasite makes a home for itself by sending proteins from the parasite into the host cell to cause a change in the shape of these cells. The human cells begin to take on the shape of a bubble and surround the parasite. And the parasite then has a home to be able to grow and divide and survive without the human immune system interrupting this process. And so for my research, I'm trying to identify these proteins that are being sent from the parasite into the human cell, such that if we find these proteins and can prevent this interaction from taking place, we can leave the parasite homeless. So to do this, I looked at the proteins of other related parasites that are sent into the human cells and tried to find common characteristics. I then tried to find some of these common characteristics in the proteins of Cryptosporidium and used these proteins as an in vitro screen. And when using this screen, I looked for a color change in my cells. And in the laboratory, a color change in the cells meant that the protein was being sent into the human cell, ultimately making a home for the parasite. And so once these proteins are identified, we can begin to prevent this interaction from taking place, again, preventing the parasite from having a home. In our world today, there's only one drug that exists that can be used to treat Cryptosporidium infection. And this drug is useful in individuals who have a healthy immune system. But those who have a weakened immune system, such as those who have HIV or AIDS or are severely malnourished, their immune system is weakened, and the drug is no longer effective. And so these people are oftentimes infected with the parasite, and these are the people who are experiencing the highest death rates. And so there's a great need to develop a treatment to be used in these individuals. And the strategy that I'm presenting today provides a great way to do so. Thank you.